Okay, so I'm here for my R34 group. We're investigating transformative optical imaging of brain and behavior in navigating genetic species. We stretched that acronym quite a bit because we wanted to land on tongs, because what's a barbecue without tongs? So um, Catherine Nagel and I are at NYU Grossman School of Medicine, Nathan Shaner's at UCSD, Andrew Strauss at Freiburg, and Jane Wang's at Cornell. We share a common goal with many of the projects here, not advancing, to understand how patterns of neural activity allow for effective navigation of complex and diverse environments. And really, really broadly, we set out to bring this team of people together to play to our strengths as we attempt to measure unconstrained behavior across evolutionarily diverse species. We're going to try and non-invasively monitor neuronal activity in freely moving animals. And ultimately, we'd like to model the ways that neurons allow behavior to adapt to the constraints of the physical environment. Let's talk about the species real quick. There are three that represented: the vinegar fly, Drosophila melanogaster, which allows really unprecedented access to particular neuronal circuits. We've got an arthropod in here, Pariali hawaiiensis, a crustacean that shares navigation circuitry with the fly, but of course has to navigate underwater, completely different physical environment, especially with respect to sensory stimuli. And then the fish, which shares the environment, the crustaceans, but has a vertebrate nervous system. Okay, so the aims of the grant, the first thing we're hoping to do is to investigate how these diverse organisms control their bodies to navigate through complex multisensory environments. And then aim two was to advance tools to measure neuronal activity during unconstrained behavior. So playing to strengths, measuring unconstrained olfactory navigation has been the bread and butter of the Nagel lab. We've developed an assay to do it in Drosophila in the upper left. You can see a pretty straightforward setup with a camera, an infrared filter, and then an arena. The arena, if we look at it from the side, has 4 columns. Each column has its own air input and can receive air and odor from a directional laminar source. And then each of those little black dots is a fly that can walk either toward or away from or in circles, depending on the stimulus. So you can see traces that show that during the odor, the fly runs upwind. That's the magenta trace. And when the odor goes off, they circle and try and find it again. And you can get probabilities of movement. You can get the velocity and the curvature. Paper in the lower right is the first from the Nagel lab, but there have been many since exploring not just the behavior, but the neuronal substrates of olfactory navigation in the fruit fly, an exceptional model for that. So underwater, odor travels in a completely different way. You know probably that many underwater creatures use olfaction to find food and navigate. And so Nagel Lab, as part of this grant, has built an optimized laminar flow chamber that will let us present olfactory stimuli both to zebrafish and to the Parhyale, the crustaceans. We've got an apparatus that lets us measure behavior with respect to gravity in Drosophila and larvae. We model this behavior. This is where Jane comes in, who's an expert in modeling behavior. Andrew's expertise is measuring neuronal activity in freely moving Drosophila. So, and then we're monitoring bioluminescent activity. You can see the photons emitted from the brains of zebrafish as they swim freely in an arena. And B gives you kind of shiny little heads of the fish. And so we're putting all this together and hope to make some progress. Thank you.